It's a pleasure to speak to you today with regards to my talk entitled Physiological Culture Media and Mitotic Euploidy with Morphology as a Second Indicator. My name is Dr. Tex VerMilyea. I'm the Vice President of Scientific Advancement for Ovation Fertility, along with the Scientific Director at Texas Fertility Centers, as well as the Director of Laboratories and Operations for Ovation Fertility in Texas. I started off my career in education as a Bachelor of Science from Cornell University, where I graduated cum laude with distinction in research. I then went off to the University of Birmingham in the UK, where I got a PhD in chromatin and gene expression. I was fortunate to be a research fellow at the Japan Society for the Promotion of Science in Kobe, Japan at the Riken Institute back in 2008. And then I got back stuck into human embryology as a research scientist and senior embryologist at Shady Grove Fertility, which at the time was the largest IVF clinic and laboratory in the United States. I had my first laboratory director position at the University of Pennsylvania in Philadelphia in 2012, and then I got recruited to go to Fertility Associates in New Zealand as their scientific director. And I'm currently now back in Texas working with Ovation Fertility, and I oversee 2 laboratories here in Texas, but also am tasked to provide the latest scientific advancements for our entire Ovation network. Here are a few of my disclosures. I am a key opinion leader and scientific advisory board for Fuji Irvine Scientific, and a collaborator for Serono, owner of Embryo LLC, scientific collaborator for Life Whisperer, as well as a partner of Ovation Fertility. So today I'm going to talk about how a change in the concentration of lactate in a common culture media, how we've been able to identify that or speculate that we feel that it provides a bit of a stress reliever on metabolic and mitotic activity within the embryo, and how that ultimately has led to a decrease in numerical chromosomal abnormalities by PGT testing of the embryo. In addition to that, I'm also going to talk about morphology and how that continues to play a role in the selection of euploid embryos for transfer. I'll start off with a little bit of history, going back to physiologist Sir John Hammond. Back in 1949, he was able to show that 8-cell mouse embryos could grow into blastocysts in a partially defined growth solution. Basically, it was components were made out of saline supplemented with a bit of egg white and yolk. And again, this is the first time to show that mammalian embryos can actually develop in a partially chemically defined solution. Then in 1957, Dr. Wesley Witten was able to further add a bit of tweaks to that culture media, and was able to show that embryos can continue to grow to the blastocyst stage. Mouse embryos can grow to the blastocyst stage and overcome the 2-cell block, where they were able to utilize embryo energy substrates like pyruvate and lactate, and how they were more readily usable than 6-carbon glucose. And that was coined Witten's medium, and basically our first culture media used within our laboratories. Now, as we're aware with the mouse embryo metabolism, early on the embryo requires lactate and pyruvate up until about the 8-cell stage, and then pyruvate uptake declines from the 8-cell stage to the blastocyst stage, but that's when glucose metabolism also increases. And at the blastocyst stage, as we are all aware, glucose is the main nutrient for provide embryo development. Now, back in 2000, Michelle Lane and Dr. Gardner were able to show how lactate regulates pyruvate in the pre-implantation embryo. And by showing that a concentration of 5 millimolar to or 0 millimolar to 5 millimolar to 10 millimolar pyruvate uptake increased while the lactate concentration decreased. Now, pyruvate uptake at the blastocyst stage significantly reduced at the lactate concentration of 5 millimolar. And the concentration of lactate, as we know, it affects pyruvate uptake. And commonly, current culture media is about 6 millimolar lactate. Now, they also were able to further show that the lactate in the medium not only altered the pyruvate uptake, but it also altered the metabolic fate of pyruvate. And under normal circumstances, the reaction of LDH, or lactate dehydrogenase, is nearly close to equilibration with oxidation and reduction. So breaking it down a little bit, at high concentrations of lactate, LDH exhibits feedback inhibition, and the rate of conversion of pyruvate to lactate is greatly decreased. Now this decrease in conversion of pyruvate to lactate further decreases the regeneration of NAD+. And as we know, glycolysis requires an oxidizing agent, which is NAD+, to enable to convert glucose to energy. So it's very complicated, somewhat like the cell cycle, but let's break it down a little bit easier for better understanding. So if we have a high concentration of lactate within a culture media, that provides a negative feedback of LDH, and therefore pyruvate does not convert to lactate, and therefore it decreases the NAD plus and NADH ratio, and this then affects the conversion of glucose to pyruvate through glycolysis, and therefore ultimately will impact the energy conversion pathway of the cells and therefore developing embryo. So there's been some very interesting studies that look at components in culture media, and one of them is by Dean Morbick from ASRM back in 2017, published in Fertility and Sterility. And he was able to show that 4 different culture mediums was able to break down concentrations of glucose, citrate, octanate, lactate, pyruvate, as well as the LP ratio. And as you can see, global culture media had 4.9 millimolar, CSC had 5.71, GTL 10.01, and 1-step 4.35. So as referred to the previous slide, around that 6 millimolar is kind of where CSC currently is, as well as global, close to that. So therefore, what happens when we drop that millimolar concentration of lactate from 6 millimolar to 1 millimolar? So what we were able to do in 2 of my laboratories, Lab A and Lab B, is we simply just changed out the culture media, whereby we were previously using a sequential culture media protocol, and instead of using a culture media sequential culture protocol, we simply swapped out the culture media and used a low lactate culture media. So for Lab A, basically composed of mostly antagonist stimulation cycles with a Lupron trigger. We previously were using a sequential culture protocol, and we transitioned to CSCM-NX, which is the low lactate, back in 2017. And we culture these embryos in a 6-6-88 culture environment, undisturbed from day 1 through day 5. And then we look at embryos on day 5 and day 6 and day 7, day 3. So we basically just check for fertilization and put them in the culture media. And this medium is an undisturbed culture system whereby we do not refresh the medium. So there's no renewal of the culture media. And then on the other hand, we had also a laboratory B, which again composed primarily of antagonist cycles. We primarily use an HCG trigger, and previously we were using a CSCM undisturbed culture media, which was the high lactate at 6 millimolar, and we transitioned to the low lactate 1 millimolar around January the 1st, 2018. Again, these embryos are cultured in 6-6-88% culture environment, undisturbed day 1 through day 3. On day 3, those embryos were removed, morphology checks were done, and ablation to the zona occurred if those embryos were due to go to embryo biopsy for PGTA analysis. But again, there was no renewal of the culture media during this time. So let's look at some of the data. So we look commonly at the blastocyst utilization rate, or the BUR, as a common KPI or key performance indicator within our laboratory. And these are any embryos that have either been transferred, frozen, or biopsy quality blastocysts as a ratio of the 2PN normally fertilized embryos. So in Lab A, whereby we went from a sequential system to a single-step low lactate media, pretty much the same. Blast utilization rate of about 43% between those two culture media systems. So really didn't see an increase or a decrease in overall blast utilization rate when we switched from sequential to the low lactate. But for Lab B, same thing, where we went from a high lactate, which is the CSCM 6 millimolar, to low lactate CSCM-NX. again, we didn't see a significant difference between the blast utilization rate between those two different culture mediums. There was reported, however, a study in California where they used the embryoscope and compared CSC complete versus the CSC NX, which is again high lactate versus low lactate. And they were able to show that, again, there was a significant increase in the number of usable blastocysts from embryos grown in the low lactate compared to embryos that were grown in the high lactate, which is at that 6 millimolar concentration. So again, you could compare perhaps, using in a closed environment as the embryoscope versus an open standard incubator. Maybe that had a difference, but they were able to show that there was a significant difference in blast utilization rate between the high lactate and low lactate, whereby the low lactate provided more usable blasts. What was interesting, however, is when we went back and looked at the aneuploidy rates of these embryos. Traditionally, in our laboratories, we do embryo biopsy and PGTA analysis for roughly 80 to 90% of all cycles. So what our data showed for Lab A is that overall there is a significant increase in the number of euploid embryos from those embryos that were biopsied and cultured in the low lactate media versus the sequential media. And that was fairly significant at p less than 0.05. Now again, we only used a cutoff of less than or equal to 30% gain or loss of chromosomal abnormalities, and so we didn't consider mosaics within this data. But in general, quite interestingly, that we were able to identify more euploid embryos in the low lactate culture media. Now we looked at Lab B when we went from a high lactate, 6 millimolar, to a low lactate, 1 millimolar concentration. We even saw a more significant increase in the number of euploid embryos obtained from those embryos that were cultured in the low lactate versus the high lactate. Now this was a bit unexpected, but because we were able to do so much embryo biopsy, it's quite interesting, of a result, and we want to obviously investigate further to see perhaps if there's a mechanism in place whereby reducing stress of the embryos is causing perhaps less chromosomal abnormalities in these potential embryos. Now, when we look at the implantation rate from Lab A, again, Lab A, we went from a sequential system to CSC NX, which is the low lactate media. We did see a significant increase in implantation success overall with those embryos that were transferred, which were previously cultured in the low lactate media. But when we compared the high lactate to low lactate, we didn't see a significant increase in implantation, although the trend does show in favor, perhaps, of the low lactate media versus the high lactate media. When we looked at delivery rates across the board, comparing the low lactate versus the high lactate versus the sequential culture system, you can see across all age groups, which are broken down. But when we look at the total, there was not a significant increase in delivery rates per transfer or per transferred embryo between the low lactate and the high lactate culture media, but we definitely did see a significant increase in delivery rates from low lactate 1 millimolar culture media versus sequential culture systems. In comparison as well, there was another publication at ESHRE in 2018 that showed the difference between low lactate and high lactate, and didn't show a statistical difference in fertilization rate, day 3 embryo development, or blastocyst development, but there did show a positive trend for clinical ongoing and implantation rate improvement with the low lactate versus the high lactate culture media. So as a total, we were able to show that there was a significant decrease in mosaic chromosomal abnormalities or anomalies between Lab A and Lab A when all we really did was change the lactate concentration in the culture media. We did not change the drug stimulation routine or the actual culture environment. All incubators stayed the same. Personnel was pretty much the same over that period of time. Stimulation drugs were not changed. The biopsy technique, which I'll talk about a little bit later as well, did not change and was fairly consistent across all operators. The PGTA platform using next-gen sequencing did not change. We have our own genetics company within Ovation Fertility, so we're able to do our own genetics, and therefore that platform didn't change, nor did our calling policy change. We didn't call embryos to be euploid or aneuploid. Those are all stayed the same during this study. Now we know there's a few types of chromosomal anomalies, which can either be numerical or structural. Now the type of chromosomal anomalies normal to mitosis, sister chromatid in the anaphase lag is lost to improper spindle formation and can therefore lead to monosomies. Non-destruction can lead to trisomies, where the sister chromatids or chromosomes fail to separate normally, as well as errors in cytokinesis, therefore alluding to abnormal chromosome segregation. So these type of anomalies could directly have an impact based on potential stresses of the embryo within the culture conditions. And so we want to definitely look more into what potentially the high lactate versus the low lactate could be encouraging some of these anomalies potentially within the embryo. Now we do know aneuploidy is the most frequent cause of chromosome missegregation within the cell and within the embryos. And temperature changes, as well as oxygen concentration and culture medium and hormonal stimulation regimes, could affect the spindle assembly and chromosomal segregation. So going back to that culture media and the influence of perhaps a higher lactate media is not as physiological as a low lactate media, therefore less stress is being endured on the embryo, and perhaps less of these chromosomal segregation anomalies potentially. And we do know that aneuploidy alters dosage of genes on affected chromosomes, and it's an overall effect of missegregation, as well as aneuploidy itself introduces certain cellular stresses, which in turn can create additional chromosomal instability within the cell. There's a great paper, it's a little bit dated now, but in 2018 in Cell, which really talks about the cellular stresses which are associated with aneuploidy. And we know that aneuploidy can lead to mitotic stress due to lagging chromosomes, as well as replication stresses, slow of replication of the fork progression, thereby adding additional stresses, and metabolic stress, the increase of reactive oxygenated species and levels in the mitochondria. And interestingly, overall, as you have aneuploidy cells can produce more ROS, which further damage DNA and therefore ultimately leads to replication and mitotic errors within that embryo. So I'm going to switch gears a little bit here, and we've talked about the implications of potentially using a low lactate culture media in the lab, and shown that we've seen and been able to identify more euploid embryos when they're grown in a low lactate culture media. But even if we have embryos that have been genetically screened and are deemed to be chromosomally normal, how do we choose that best embryo, or how do we choose which embryo to transfer, which one's the best? And we traditionally have defaulted to morphology of that embryo as a second indicator. So we screen the embryos, we identify which ones are euploid, and then we go back and look at, what does that embryo look like? And a lot of that is, very, very subjective, and there's not an objective way to really look at embryos. Morphology is all we have currently at the moment, other than morphokinetics and artificial intelligence, which I'll talk about a little bit later. But we traditionally use commonly across the globe morphology. And the point of this slide is really to illustrate that it's really the eye of the beholder, or what someone really sees. So these are common franchise brands within the US, and I'm sure some in Japan. I do remember Baskin-Robbins in Kobe a couple of times. But interestingly, with the FedEx sign, I don't know if you've ever noticed, but there's a little arrow within the word FedEx, and it's supposed to mean speed and precision with regards to delivery. Now Amazon, Of course, there's a smiley arrow, and that's because they sell everything from A to Z. Took me a while to realize that. Now, Baskin-Robbins, if you really look close with their new logo, you've got a 3-1, and that's because they're notoriously known for providing 31 flavors of ice cream. And Tostitos, which is a brand of tortilla chips in the US. Right in the middle, you see two people kind of having a bit of a party over a bag or over a chip, and looks like some salsa of types. So again, this slide is just to illustrate that even though I may be looking at an embryo and think that it's a high-grade embryo, someone else may come along and think that it's a different grade of an embryo, and maybe not as high or perhaps lower than what I would have graded. So it'd be nice to have some more objectivity around embryo assessment. Now, previously we looked at embryos to sort of get a gauge of morphological identification and morphological selection, and a lot of time was spent early on with pro-nuclei grading. And then time-lapse, we were able to kind of use a little bit of nucleoli grading and provide some evidence that potentially the positions of the nucleoli within the nucleus or pronuclei was able to distinguish or identify which embryos may grow on to be better developed or lead to a viable pregnancy. And then we also took a snapshot around day 3, looking at fragmentation of the embryos, and the thought there that a lot of fragmentation would lead to a not so good-looking embryo, or perhaps decrease that pregnancy success of that particular embryo based on the degree of fragmentation. But really, our laboratories have gotten so good at growing embryos to the blastocyst stage that's really where we look now. We look at the blastocysts and put these blastocysts in scales or in grading ranges, categories based on the size and shape of the inner cell mass, as well as how many cells in the trophectoderm and how expanded these embryos are, so on and so forth. So blastocyst grading is kind of that additional indicator based on morphology as to which embryo to transfer. Now, if we were going to have a live presentation, I was going to poll the audience or ask these questions, but I'll still ask them and give you a bit of time to think about it. But I'm curious to know about morphological assessment of embryos every day. Is your laboratory a lab whereby you take the embryos out of the incubator, or even if they're in time lapse, do you look every day to assess the embryo's morphology and record it accordingly? Secondly, what about just assessing fertilization on day 1 followed by blastocyst grading? As I showed you in lab A, after fertilization check, we put all 2 PNs or normally fertilized embryos in a culture dish, and then we put them back in the incubator, and we didn't open up the incubator until day 5. Therefore, we were not taking embryos out on day 3 or assessing, but we have so much confidence in our culture system that we think that we have a very, very good environment to replicate that of the uterus and the uterine environment to grow those embryos to the blastocyst stage. So curious if there's anybody who just does a FERT check and then doesn't open the incubator until day 5. Thirdly, what if you use time-lapse, but you only assess morphology on certain days? You could push record and start taking images immediately after ICSI or perhaps after day 1 at fertilization check. And if pictures are taken every 5 minutes, you may only come up to the time-lapse viewer and watch the movies only on day 3 and then again on day 5. Or do you sit there and take morphological assessments through the continuation of the entire video in the embryo growth stage? What about using day 3 morphology to decide if those embryos are going to be in a PGT cycle? We have some laboratories that, depending on if the embryos are grade or good grades or 8 cells on day 3, that may dictate whether or not those embryos are going to continue to get cultured to the blastocyst stage for potential biopsy. Or if the embryos are highly fragmented, they may choose just to do a freeze-all cycle, or even perhaps do a fresh embryo transfer on day 3. But curious if people still use day 3 morphology to decide whether or not those embryos are going to be cultured to a PGT cycle. And lastly, what about those laboratories that are under the impression that, as long as you have a blastocyst to biopsy, it doesn't really matter how that embryo got to the blastocyst stage. We're going to, freeze a good quality embryo. We have to blast and we have to biopsy a good quality blastocyst. So really doesn't matter the previous 5 days, just as long as it got to the blastocyst. And we'll have genetic analysis assessment to identify whether or not that embryo is chromosomally normal or not. So, the end point is just having a blastocyst at the end of the cycle. Now, I'm sure we're all very familiar with the Gardner grading system, and of course it's a 3-part grading system which looks at the expansion of the embryo followed by the inner cell mass and the trophectoderm. But there's a constant debate as to, based on morphology, which component of the composite score has the greatest impact on IVF outcomes. Should we be focusing on that embryo that's got a great trophectoderm, or should we be focusing on the inner cell mass, a nice clumping of cells for the inner cell mass, or if we have too few cells in the trophectoderm, does that mean that the embryo won't even implant, but we have a nice healthy inner cell mass? So I think it's very still debatable within the field as to which component of that score, or even expansion, doesn't matter if the embryo is fully expanded or not, but focus on, of those 3 components, expansion, inner cell mass, and trophectoderm, which one is more likely to lead to a successful. Now, interestingly, there's been proposals of having a numeric alpha grade, whereby the expansion grade is numeric 1 through 6, but as we continue to culture embryos better within our laboratories, and some labs culture embryos all the way to day 7, we have embryos that are completely hatched out of the shell, as well as if embryos, the zona was breached on day 3 for embryo biopsy, usually by day 6, late day 6, some of those embryos may have completely hatched out of the zona pellucida. So as culture environment has improved, our grading spectrum has increased. So we've had to go out to that expansion grade and probably have more day or more expansion grade 6s because those embryos have completely hatched out of the shell. Now, the Society for Assisted Reproductive Technologies in the U.S. has tried to implement a national embryo grading standard, which is more of a descriptive scale grade. Again, good, fair, and poor based on cleavage or morula or blastocyst stage. And that then goes based on expansion, as well as the inner cell mass and the trophectoderm. However, alpha has really sort of encouraged a numeric scale grade universally or globally to an international move for subjective to a more objective grading, whereby it's a combination of a grade and rating based on expansion as well as inner cell mass and trophectoderm, focusing more on a numeric scale grade. But the problems with all this is that any morphological assessment is very static and it's operator dependent. And unfortunately, there's no standardization of morphological grade suitable for blastocyst biopsy. So we tend to have a personal interpretation as to which embryos are more likely suitable for biopsy based on how they look. But again, your colleague, even though we try to be consistent on our embryo grading, it may be that they may have a different grade of an embryo, and therefore that embryo may not get biopsied or frozen based on a lower grade given to that embryo. So there's been multiple studies which really look at, again, what should we be focusing on? Is it the expansion of the embryo, or is it the inner cell mass grade? But looking at euploid embryos which are chromosomally normal, one paper states that the inner cell mass was the best predictor of sustained implantation. When you have a cohort of chromosomally normal embryos, that you should really focus on the inner cell mass grade. But the trophectoderm grade of an A or a B compared to a C had significantly higher likelihood of ongoing pregnancies or live birth. So if you had a trophectoderm grade of a C, that would mainly not lead to a pregnancy, therefore focusing on trophectoderm grades of an A and a B. And then following by expansion, grade of a 4 or 5 compared to a 6 had significantly higher likelihood in ongoing pregnancies and live births. We've seen this also in our laboratories, whereby our completely hatched blastocysts, even on day 6, have a lower pregnancy success than those on, than those that are still retained somewhat within the zona pellucida. Now, when we looked at our data, comparing non-fully hatched blastocysts with an expansion grade of 5 versus fully hatched blastocysts, we were able to show that non-fully hatched blasts had not only a higher initial pregnancy and SAC, but overall positive fetal cardiac activity. So there's something perhaps integral with regards to the retainment of that embryo within the zona pellucida to provide perhaps additional pregnancy success. Now, when we look again at additional embryos that have been chromosomally tested, embryos with the highest score showed higher euploid rate. So those embryos that were graded on the higher spectrum, based on this paper published in 2014, show that indeed the embryos with the highest score showed higher euploid rates. So something to be said about better good-looking embryos perhaps are more chromosomally normal. What I found interesting, though, is the complex aneuploidy correlated with embryo morphology, and therefore those embryos that were more on the poor scale with regards to perhaps more fragmentation or poor cellular development, those tended to lead to more complex aneuploidies. And which is a little bit reassuring is that faster growing embryos showed a similar euploid rate as slower embryos. And the idea that perhaps some embryos that are in a single-step culture media may grow a little bit faster. There was initially some concern that perhaps those embryos were potentially more chromosomally abnormal, but this data in this study show that faster growing embryos showed a very similar euploid rate than those embryos that grow a bit slower. So to start rounding around the talk here and finishing up, is looking good still important? So can we still continue to put quite a bit of emphasis on the embryo's morphology and how they, how those embryos look? And we obviously realize that there's a lot of subjectivity, and it'd be great to remove that and make embryo assessment more objective. Even to a way that we're not having to rely on genetic testing, and we can use tools and have tools to help us identify which embryo is going to be superior based on just the embryo's morphology. And one way to be able to do that is by using artificial intelligence. And I've had some collaborations with Life Whisperer, whereby we've been able to use deep learning and computer vision to further help us identify which embryos are more likely to lead to a pregnancy. And obviously, as we know, artificial intelligence can pick through things a lot easier and identify morphological features within embryos a lot more accurate than we can with the human eye. And as we know, the deep learning handles unknown patterns and features which are not visible to the human eye. And again, it's able to make correlations in different parts of the image to help piece together a better identification of whether or not that embryo is potentially more viable. And then you layer on that with computer vision and focusing in on key points of interest within the embryo and assessing features within a region, as well as using automatic image segmentation of that particular embryo, all in an automated way. And the beauty of this particular system that we have used is it only takes a static 2D photo, so I don't need high complex and expensive time-lapse technology. I can use our bulk standard embryo or microscope camera to just take a static photo of the 2D image, and then that image is loaded into the cloud, and then we have a confidence level score provided through the algorithm and through the AI, identifying which embryo has the highest potential for viability success. And overall, when we compared, at least the embryologist identification of which embryo is more viable based on subjective morphological grading, when we compared that with what the AI was able to identify, we saw about a 25% improvement in accuracy of the AI technology over that of standard morphology per embryologist. And we did use clinical pregnancy as an endpoint with positive fetal cardiac activity. And we used a lot of thousands and thousands of photos to not only build and train the AI, but also to assess and really put the AI algorithm to test to provide reliable outcomes and results. So very, very exciting. It's neat to see this technology move forward. Of course, we're all very aware of again, cell-free DNA amplification and non-invasive PGTA analysis, which I feel, again, is going to provide a more potentially accurate assessment of embryo genetic competency. But again, providing a bit more of an objective assessment of embryo viability and success versus what we currently routinely use across the globe, which is that subjective embryo morphology grading scheme. So in summary, and I'd just like to close with a few points, it shows that early on, our data shows that physiological culture medium correlates with a decrease in chromosomal anomalies and an increase in live birth rates. Now, we're still trying to investigate the mechanism, if indeed there is one that is causing these embryos to be more chromosomally normal by being grown perhaps in a more physiological low lactate culture media. There's still some very interesting studies that we'd like to do, but preliminary data shows that not only are we getting more euploid embryos, but ultimately are obtaining more live birth outcomes, and you would assume that would go hand in hand based on transferring embryos that are more chromosomally normal. I do think that standard morphological grading still has value in our daily workflow within the laboratory. Until we get systems in place and reliable systems like artificial intelligence or perhaps non-invasive, cell-free DNA assessment through spent culture media, I think we are still going to go back and use morphology as an indicator of some sort within that workflow. We all like to see things that are pretty, and we don't know the textbook blastocyst is actually what a blastocyst should look like in vivo. So I think we'll always have a place of some sort with regards to referring or deferring to embryo assessment and morphology. And I'd just like to also give a quick plug on the biopsy technique, and I really feel that this is critical to success. We've done some internal studies that show that if you use too many, laser ablation pulses, as well as too rough with the embryo biopsy technique, that you can actually induce, or you can cause those cells to provide a aneuploid diagnosis for that particular embryo, or increases in mosaicism. So I strongly encourage those who do a lot of embryo biopsy to sit down with your fellow colleagues, try to objectively find a technique that everyone can use, and try to just be on the same page. Look at ploidy results per technician. See if a technician, embryo biopsy scientist or embryologist, if they happen to have more mosaic calls than the other one, then sit down and look at the embryo biopsy technique to see if there are any differences. And lastly, as I mentioned, objective embryo deselection is soon to come. I think a lot of these technologies will allow us to deselect which embryos to continue to culture or to continue to freeze and biopsy. And if you could run perhaps images of embryos through artificial intelligence and identify which embryos are more viable, and then focus on those embryos to then do genetic analysis, that would obviously decrease the cost of the IVF process, as well as speed up the workflow, and perhaps provide us with better outcomes overall. So with that, I appreciate your attention. I wish I was able to give this talk live in Japan, But hopefully next year. And yeah, happy to assist or help with any questions that you may have. And I hope you found it educational. And we'll see you next time. Thank you.